Okay, then ready to start. I'm going to be talking about the rat model, maybe adding a few elements of our own experience to the model that Sully presented very well. Then I'm going to be talking about gene expression analysis, especially with a focus on pathway analysis and gene network regulation in these rats in comparison to human profiling. So the model. We heard from Sully there are multiple models of animal models of HIV. Most transgenic express one protein. Then there are infectious models. I'm not going to get into detail. They have a number of problems in the rodent, including, well, for the experiments that we do, they are difficult to handle. But in addition, they express a lot of proteins, but they're difficult to manage pathogenic courses, and the rodent cells are not infectable. And then there is the rat that we use and a corresponding mouse. And like Sully said, it's GAG-POL deleted, so it's not infectious. And in the rat, expresses through the LTR in disease-relevant cells. So that's very important. There is a corresponding mouse, like Sully was saying, that one has ectopic expressions due to TAT not being functional in the mouse. So, like I said, expression in disease-relevant cells. This is TAT expression detected in microglia. This is GP120 detected in GFAP-positive cells. Pathologically, they have a very consistent astrogliosis, mild but significant, and a very mild but significant and consistent microgliosis. Relative expression of relevant genes like GFAP, EBA1, and CD11B, it's all increased in these mice, in these rats. From the viewpoint of functional consequence of the expression of multiple HIV protein at low level, like Sully was saying, we see an impairment in long-term potentiation. Well, long-term potentiation, for those that are not neuroscientists, it's a form of plasticity whereby synapses become stronger. And this is, has been shown, including by us, with single glycoproteins, single viral proteins in the past. So low-level expression also recapitulate that phenomenon. Working memory, this is a paradigm of spontaneous alternation. It's a working memory paradigm that depends on prefrontal cortex and hippocampal. And the purpose here is just to show that the gene expression that I'm going to show you is at the time point whereby there is a cognitive impairment. Multiple other studies have addressed the cognitive consequences in these rats, including Sully was showing working memory in a different paradigm. So in conclusion, multiple HIV protein in this relevant cell expressed at low level, which, like Dr. Chang was saying, we like very much as a model of hand in the post-CAR era, like you proposed. Mild astrocytosis, mild microgliosis, also mild, impaired LTP, working memory. Limitation is they are constitutively induced. They are not. They have a progression, but they are not inducible. So gene expression, pathway analysis, and gene network analysis. I'm going to first describe the approach that we use, which is a systems biology approach. There are many ways to analyze pathways. Some you may have seen in previous presentation, like the ingenuity pathway. We use the gene set enrichment analysis I'm going to describe in a second. Maybe this is not yet systems biology. We also use a systems biology analysis, which means to reconstruct a gene regulatory network and interrogate the entire transcriptome at once. And we do an interactome reconstruction with an algorithm called Arachne, developed by a collaborator of ours, Andrea Califano, at Columbia. And then we interrogate it with a strategy with an algorithm called Marina, which identifies the drivers of those gene expression changes in the data set. So gene set enrichment analysis, I'm going to spend a second because I'm going to be showing a lot of these plots. What this approach does is to compare, in essence, two distributions. Let's say that this is the distribution of gene expression in a certain data set, and this is a subset. This could be a pathway, or this could be the genes that are expressed by a group. And this is another one. So you can see this one has the same distribution, although this is a subset, and this one has a skewed distribution, so this one is different. That's the essence of this analysis, and is based on the KS statistic or other statistics. This is the one we use. So I'm going to show a lot of plots that look like this. A skewed distribution, I said, means that these genes that I'm using to interrogate the data set are enriched, are expressed more in this class as opposed to this. I'll be showing graphs like this. These are genes related to mitotic activity, DNA synthesis, mitotic-related genes. And they are increased, as you see the hump on this side, in the HIV transgenic rat as opposed to the controls. So when you see a graph like this, now you know how to interpret it. So the gene network reconstruction is based on a mutual information approach that in essence finds correlation between transcription factor and candidate targets. It works over a very large and complex experimental design, whereby it's going to weed out random association and find the strong systematic association through mutual information. So high information between a transcription factor and potential target is going to interpret it as a potential regulation, and low information will be low mutual information, not a candidate for regulation. The other key mechanism through which we do the gene network reconstruction is the data processing inequality. This is a theorem of information theory that says that direct information is stronger than indirect information. Simple as that. So a transcription factor will have a target, that will have a target. Okay, it's not necessarily the case, but sometimes the transcription factor will regulate something, could be another transcription factor, regulate another gene. So this is the way you determine that this is the direct target and this is the indirect target. So this algorithm does this. So first computes pairwise mutual information. We'll have a strong statistical cutoff. Apply the DPI to remove the indirect regulations. And progressively, we'll end up with the statistical direct regulatory relationship. The type of results look like this. This is a rank of candidate master regulators, so genes that regulate a gene signature, a set of genes that changes in your condition. Each of these is one of these plots. And what happens here is that the activity of the transcription factor, say this, the glucocorticoreceptor, it's ranked by his activity, which is determined from the activity of its targets derived from the gene network analysis. Not by its expression, not by its expression, because the expression may not change. For instance, this one slightly changes the glucocorticoreceptor because it's regulated post-translationally a lot. So activity and not expression, and this is an example of validation. So for instance, the glucocorticoreceptor in this system was then down-regulated with the siRNA, as you can see, and again, by applying that type of analysis, we can tell that the predicted targets were actually let's say, dysregulated by downregulating NR3C1 glucocorticoreceptor. So the systems biology, I can define it, but most people like this definition as an interrogation of a system as a whole rather than one gene at a time. The goal of our approach is to identify testable hypotheses from the high-throughput data. Key to this approach is also this large multivariate experimental design, because in essence, this is a way to substitute for a mutation of like each and every, say, yeast cell in a culture or other approaches that were used that don't apply obviously to rodent. So to the data now, pathway analysis and gene network analysis. These are HIV transgenic rats, and these are pathway analysis done by GSCA. This is using this reactome. What they are gene sets that you see here, predetermined to be representative of certain pathways. There is a large collection in the Broad Institute. These are representative results. So big changes in inflammation. Here I have one of the biggest results, which is interferon alpha-beta signaling, which we heard before, I think was Lynn. Another one is trophic responses, which also Sully was saying. There is a reactome called neuronal system that contains a lot of trophic signaling, a lot of P3 kinase pathway, a lot of mitochondria, and this one is strongly downregulated. These are reactome with some overlap, similar stories. These are axon guidance. And then another reactome that was identified, actually two, is Parkinson's disease, and the other was Huntington disease. So neurodegeneration, you use the word neurodegeneration also, and I think the reason why it pulls them here is that they are full of mitochondrially related genes. So these are representative genes from various pathways. Prostaglandin PGD2 synthase that we heard about yesterday involve also in astrocytosis, a big change in gene. Maybe we could get some advice on some inhibitor. ISG15, interferon-regulated gene that was also proposed as a marker of progression, and so on and so forth. I'm not going to take you through all. I'm just going to mention these last two genes because they are known to be one binding to VPU, an HIV protein, the other to TAT, which tells you that probably these are effects that we see because we have all those proteins being expressed and not, say, just GP120. So comparison with human data set. This is the data set we use from the National NeuroAIDS Tissue Consortium, NNTC. It's a data set of gene expression from 24 human subjects. They include uninfected controls, infected with HIV but without significant neurocognitive impairment, infected with significant neurocognitive impairment but without encephalitis, and then individual with cognitive impairment and encephalitis. They've been processed for RNA and Affymetrix microarrays. So first level of comparison through GSEA, simply running the same analysis. What's in common? Well, in common there is a lot. These are some representative results from the rat on the top row. You saw this was the interferon signaling. This is the neuronal system, so general trophic. And this is the Parkinson's disease, so we said degeneration. And we find the same pretty much in the human data set. This is the frontal cortex on the second row, third row, basal ganglia. See, similar pattern. So overall, a lot of communality. Now, this is a gene network analysis. Here, what we've done, we use the Arachne algorithm to generate the network, and we use the Marina to interrogate it. But our transgenic rat dataset is very small, and the NNTC, which is 75 arrays, is also borderline small. So we did analyze this one, but it doesn't reach the statistical power that we really would like. So we used another approach. We use a human frontal cortex interactome generated this way that is much larger, and we interrogated it with the differentially expressed genes from the rats and the NNTC. So we found a number of candidate master regulators. These are the ones that are really extremely high ranking, which means very significant, very robust. These data are very new. I'm just going to give you a bird's eye view and a couple of examples. So these are changes in master regulators. So imagine each of these is a transcription factor that regulates a significantly large number of genes. Here the transition in the NNTC from control to HIV. Here from HIV to hand, neurocognitive impairment. Here from HIV with hand to encephalitis. This is gray matter, so frontal cortex and basal ganglia. These are frontal cortex. The last one is the basal ganglia one. And just a superficial analysis, some go up, the red, some go down. But most of the big changes happen at the initial transition between control and getting infected. The second transition, a lot less activity. The third transition, predictably, from having a cognitive impairment to having encephalitis, is another huge transition in the gray matter. So big change, small changes, big change. Obviously, here there could be the one key gene for the pathogenesis, but as a whole, not a massive change in gene expression at the transition to non-cognitive impairment to cognitive impairment. These are two that I'm going to show you next. They are the top ranking two. Conversely, again, superficial analysis in the white matter. Really, it would seem that the biggest transition is in the middle. It's between the HIV infection without cognitive impairment to with cognitive impairment, which is reminiscent, this again, wild matter, reminiscent of what Rodrigo's husband was saying yesterday regarding his patients, but again, at a very superficial level. Those two top-ranking guys in the prefrontal cortex, actually it was frontal cortex, and in the basal ganglia in humans. Two comments. Number one, this is the guy that was the top ranking in the cortex. They are both, this one especially, regulating a large number of genes. So maybe the way we did the analysis, which has a ton of statistical cutoff in between, selected really very robust transcription factors. This one has some literature before that identified as possibly being involved in mitochondrial trophies, which would be very relevant. And this is the other changer, the other top candidate, again, a very large number of genes. The activity of both is decreased. Now, I'm a little confusing you because now the graph is a little different. Here, to analyze them, we separate the genes that they control, they go up, from those that go down. Certain transcription factor activating and inhibit genes. So, I give you the essence. The ones that are typically increased by the transcription factor are less increased. The ones that are decreased are less decreased because they were decreased, both of them in HIV. Same deal with the rats, same profiles. Not all of these are as significant because this data set is considerably smaller than the human. So, moral of the story, I'm going to draw some conclusions and then I'm done. Gene expression changes. Pathway consistent with the astrogliosis and microgliosis that we observed, and that I'm sure that Sully observed also. Increased inflammatory related genes, like Sully found, predominantly what jumps at you are interferon regulated genes. I mentioned ISG15, a marker of progression according to some, according to one of our collaborators. So impaired trophy support, again, similar finding that Sully found. And neurodegeneration, again, like Sully also observed. Change in phospholipid metabolism, I didn't discuss it. Change in mitochondria quite a bit. Peroxisome, I didn't discuss it, but these are also prominent changes. Last conclusion, so the gene network approach, first of all, before that, pathways largely consistent between human and the rat. So this supports the validity of the model. Number two, gene network analysis, in part I say because it's just the beginning of an analysis, but certainly point in that direction because the gene regulatory logic may not be exactly overlapping in different species, but this method will identify the overlapping ones, and there are some that seem extremely robust, at least at the computational level. Gene regulatory networks also gives us those testable hypotheses, because now we have some potential targets to probe. And here I'm done. The next steps with like a very large data set from the rats, which we are hoping to get funded to, like everybody else, to generate, because obviously it would be nice to do the gene network analysis in the rat at the same type of resolution as the gene network that we have in the human. So here are the people that participated in the study. Almost all that I showed you are papers where West Canonico is the first author. Lena van der Stap did a lot of the analysis, but most of the analysis was done by Andrea Califano, and then people that did various things. Oh, obviously, Elijah Masnia did the pathology, George Kub, Olivier George, Marisela Morales did various things, Marisela did some morphology, and George did help with some of the behavior.
Thank you so much for keep the time. And you use up your 20 minutes. So I think probably we can hold just one quick question, very quick question. This is the 30 minutes, 30 seconds. Okay.
No, questo basta bene.